So what I'm going to talk about this afternoon are some of the research studies that we did at Rosalind Franklin University. And I'm going to break this up into two parts. You're going to get some of it this afternoon and then the rest of it tomorrow morning. So just a little background of how this all started for me and our research at Rosalind Franklin University. In 2003, I investigated the applicability of using the LMD for forensic use as just a thesis topic. So it actually just started kind of as a little small project. And then in January of 2004, myself and Dr. Peterson at the lab, we wrote an NIJ grant proposal to further investigate this technology. And then in early 2004, we presented the AFS presentation of the preliminary data, which there was a lot of interest for, and we were excited. And by June 2004, we were awarded the grant, and then a 2-year grant to investigate this. Several presentations that we've made that some of you might have seen throughout the last couple of years, and we published our first paper in July 2006. And here we are, 2007, and we're doing, the grant is now over and complete, and the final report is available on the web. It's this gigantic 100-page report. So all the stuff that you will see me talk about, it's described actually in more detail in the report. Or if you forget, or if you have a question, you might want to refer to that. And then right now, there's a second manuscript in progress, because essentially the first manuscript was just the first half of the research. So just, you saw this slide before. There are different separation methods, obviously. Preferential lysis, flow cytometry, microchip separation, membrane filtration, magnetic antibodies, microbenzone, and we are doing laser microdissection. So the studies that I will go over that we thought were important in terms of determining whether LMD was going to be feasible for cell separation of sperm cells from epithelial cells are listed here. And the first one is a histological staining study. So we want to determine the stains that we were using on our slides. How are they going to affect our downstream analysis? That's important. Something that you before never really had to deal with unless maybe you had an old archival slide that was stained and you were going to swab that and try to do DNA analysis. And then the DNA isolation study. What type of isolation method is best suited for these types of samples, especially since they're stained? And also, in what kind of format, because the sample is obviously collected in a certain way, and what's the best way to isolate these cells. We also did a yield evaluation of qPCR. I'll, of course, describe that in more detail, but we did qPCR on the cells we collected to determine what our yield was. Then, finally, about halfway through, we actually start determining whether it can separate a mixture through our mixture separation study. And that also folded into our low copy number study, also using mixtures at the low copy number level. We then did a comparative study using the differential extraction and laser microdissection to see, evaluate both the ability to separate and detect the sperm donor. And then we were able to get a couple of cases to do a case study. So I kind of want to open this act before I go into the studies, now that you've worked on the instrument a little bit, talk about some of the technical obstacles maybe you experienced and that we've experienced using the instrument. So first of all, there's optimizing LMD cutting parameters. We didn't really go this into detail in the lab because I didn't really want to go into the software too much. Each instrument is going to be different, the way you have to optimize it. But what you might have noticed is that, for instance, in my group at least, I might have been adjusting the power to basically be able to cut through a sample. So that's something that you will have to determine actually in your own laboratory. So one of the other technical obstacles, we've talked about this, the microscope slides. They're different than anything that you've probably seen before. You have to deal with the architecture of the slides themselves and the fact that you also do not have a cover slip. Now, environmental conditions of instrument. On my side, if you noticed, I actually had a little vaporizer. We didn't have this for the first class. We had problems with static, which is on this list of obstacles. I know Kelly in her lab, that was always a concern. You had static, a lot of static in your samples. And what we've determined is that you have to keep it at a certain humidity in the environment around the instrument. My lab, the instrument was in a small room. In Kelly and Pat's lab also, I think originally you had it in a large lab, but then you moved it to a small room.
Yeah, originally we had it in a pretty large room. How big is that room, Pat? Probably. The big one? Yeah, the serology lab. Well, about 20 by 25 feet. 20 by 25 feet. So it was very large, and then we moved it to a smaller room in the basement, which is really, really nice for humidity, of course, in Louisiana. So that's probably an 8 by 8 room, and that is so much nicer. I mean, and just to be able to control the humidity is so much easier there. And the light.
Right.
Yeah.
So that's one thing that you'll have to consider is where you're going to put this instrument, because environmental conditions can be critical. We were getting a lot of static a couple days ago. It wasn't so as bad today. I don't know if it's all the rain or something, but even the air conditioning, as I think the day goes on, the air conditioning is, starts drying the air more, because in the morning it was a little more humid on your side, and then it was drier later.
It didn't seem too bad. I did notice, of course, as we went on, it seemed like it got a little worse, but it doesn't seem as bad as when we were doing it with the other class.
Okay.
And just going back to the room thing, I would highly recommend any of your agencies, if you've considered getting a microscope, if you have a microscope, of course, you want to make sure it's in a smaller room if you can. But otherwise, if you don't have a microscope yet, that would be a big recommendation, I think, is try to make it its own room. Plus, these microscopes, they're very sensitive, so.
Yeah, so the environmental conditions is that you want a pretty good humidity. Now, in Chicago, where we had our instrument, my hydrometer, it seemed that I cut well when I was about 35, 45%, which seems kind of dry compared to you had more ideal cutting closer to 60.
Like 50 to 65%, I found my range was good.
Right.
So I'm not sure.
You'll have to see what's the best cutting environment. But the idea is that you don't want it too dry, because that's bad for static. The opposite is that you do want your slides themselves to be dry. So don't think that if you pump it up to 80% humidity that you're going to get a better cut, because then your slides will probably absorb some moisture, and it'll be a little harder to cut. That's why I like to put it in the vacuum desiccator and pull them right out and then start cutting. I have nice dry slides. In the Chicago, early on in the Chicago summers where it was just very humid, if I had my slides laying out like all day because I was like waiting to cut them, they actually would cut not as well as if I had cut them in the morning. Something to consider. So there were some things that you probably all saw were the hanging chads, where it wouldn't quite cut all the way down, and you would see a little hang, the film and the cell hanging down below. Now it's pretty easy to kind of basically cut those out using that move and cut feature. I found on the older instrument I'd have a higher rate of hanging chads, and probably even today, this week, because I don't have the instrument completely optimized exactly the way I want it, I might have a higher rate of hanging chads. You really have to balance the power and speed and things like that to get a good cut. But I think it did. I know. How did you guys feel about how easy it was to knock out the chads? Is it okay? And again, we just talked about static being an issue. Static, now sometimes if there's a lot of static and you cut out a cutting, and it might stick to the underside of the slide. It might actually fully cut out but stick to the underside of the slide, and that's where you kind of run into trouble, because you may not necessarily see it because it may go right outside the field of view. Is that correct? Is what you've experienced?
Yeah, that's why static could be an issue, because technically you may be losing sample that you don't realize you're losing. See, that's where you can lose sample in this type of procedure. One thing that I could probably go into tomorrow a little bit, but with our caps, we have domed caps that we use, so they hold a larger volume, and the engineer explained to me that, look, the more plastic exposed, the more you're going to have static. So I actually went to a larger volume in the cap, which did help a lot. So between making sure your humidity is fairly good and having the nice volume in there, in whichever type of cap you use, it might be different, but that did seem to cut down on the static a lot. And then, of course, looking in that tube cap can help you too. We did that a couple times today, and we could see, okay, we have about 30 cuts that we made, so that helps. That inspection helps a lot.
Yeah, if you didn't hear us talk about it before, The tubes I was using were the flat bottom tubes, and I was putting about 23 microliters in my protocol. Kelly's, they have the dome cap, and they put about 50 microliters. Yes. But oddly enough, these particular PCR flat top tubes somehow filled a little more, had more volume than what I was accustomed to. That's why we switched to 25 microliters in this class. And then there's also the collection buffer. You have to decide, what are you going to collect your samples, and depending on really how are you going to extract. We extracted today in Lyse and Go, and the protocol's in there that we performed out for our research. Kelly does it in TE, and she'll go over some of her protocols. The other thing is, especially with my volume, small volume, and using 23 or 25 microliters, if you take a long time to collect a sample, or if you decide to walk away for lunch or something like that, 25 microliters can evaporate, especially, again, if your room is too dry. So something to consider. You want to do an efficient collection. And so I wanted to just kind of open up if there were any other technical issues of concern. Now, I think the one thing was that tube holder. And we all kind of talked about it, that, it should be designed differently. And when my last group saw the old tube holder, too. Who saw the old tube holder? And what did you think about the old tube holder?
It looked like it would be easier.
Yeah, I thought so too. So Andy, you said that kind of Leica knows this, and
I've taken all your comments, and it's interesting. And yes, it's nothing that you've seen we don't know, and we'll certainly look at improving some of these parameters.
Okay.
And I did one more final thing. Christine is talking about environmental factors. I want to make sure that you understand is that every system has different environmental issues. So don't take what she said here that's going to make them apply to every other system. I mean, you're going to be aware that everybody else's system is going to have their own environmental issues. Okay?
Okay, yeah, that's good to know. So let me talk first about the histological study. So, okay, you can identify cells that are not stained. Pat talked about DIC, although it's difficult with the pen slides because you have that architecture, and DIC actually makes it even more enhanced, those pores, or I don't know if they're bubbles or pores. On the left, sperm that's not stained, and the right, E cells that are not stained. And I actually tried to collect sperm from mixed samples of unstained specimens. I did have an error rate of mixture sometimes when I would try to do that, because I somehow would miss sometimes an E cell nuclei I didn't see. So my recommendation is that you do need to stain these specimens. So what do we want to stain it with? Well, we want to be able to visually discriminate the sperm and epithelial cells. And then we also want to determine how is that going to affect our downstream analysis. So the part one, we actually did 2 parts, because after part 1, we found out we wanted to do more work on the staining. The part 1 study tested hematoxylin and eosin, which is a very common pathology stain and is used a great deal in laser microdissection outside of forensics. Christmas tree stain, a lot of you are using it to identify sperm cells. And a couple others, acridine orange in the literature has been used before to identify sperm. Then a couple others, right stain, which is used many times for blood smears, and also methyl green, which is a nuclear stain. And we wanted to evaluate those. One, can we ID the sperm with these stains? And then we collected cells by LMD from slides stained with these stains, and then isolated using this QIAGEN-QIAMP extraction. This is early on in our study, and this is when we were using a QIAGEN-QIAMP extraction procedure. And then we ran STR's Profiler Plus. There's just a couple of pictures. Acridine orange on the left, the non-stained, Christmas tree stain, and hematoxylin eosin of the cells. Oh, and I don't show right stain. Okay, the reason why I don't show the right stain is because actually somehow that stain caused some deformity in the pen film. I have no idea why, and I didn't really investigate any further, but it actually made it very difficult to identify the sperm. And in addition, the methyl green, I don't show up there either. Methyl green, although it's just a nuclear stain, it's a very hard stain to control intensity. So it was difficult to work with. So this is just kind of an estimate of the rating of how easy was it to identify sperm cells and epithelial cells using these different stains. And be aware, this is just one analyst, me, doing this, so there's a bias. But it's kind of given a general idea of, is this stain going to work or is it not going to work? And obviously the pluses mean good and the minuses are bad. And you can see CTS, the Christmas tree stain, performed the best. But there is a bias because I have 10 years experience doing Christmas tree stain. Hematoxylin eosin I don't, but if you go to the UK where they primarily use hematoxylin eosin for sperm identification, I bet if I gave them the slides, I wouldn't be surprised if those scores were reversed. So really, H&E or Christmas tree stain were good stains to identify your cells. That's no big surprise, really. So let's look at hematoxylin eosin and the Christmas tree stain. What we did is we ran the STRs on them. And what this chart shows is the RFU values that we obtain from stain cells normalized to unstaged specimens. So the hematoxylin eosin, the RFU values were about 62% less than what unstained specimens. And these are not mixtures. These are we're just doing sperm smears. And then with the Christmas tree stain, that was even a little less, 43%. And then we determined that actually those stained specimens exhibited RFU values significantly lower than unstaged specimens. So there is an effect there. So in summary on the part 1, okay, I told you methyl green, right strain, not suitable for LMD, mostly because of ID. And cell stain with acridine orange resulted in no amplified product. It is an interchelator, so that's probably the reason why. And Christmas tree stain and hematoxylin eosin, they're both good for sperm ID, but there are significantly lower RFU values than the unstained control. What are we going to do? Oh, the thing, though, is even though they were low RFU values, when we collected these large amounts, 300 sperm or 150 epithelial cells, we still got results, still got genotyping results. So that was promising, even though the values were lower. So those results prompted us, let's see if there was another, okay. But we did determine at this point that H&E was the best choice. It performed the best. But we wanted to look into this further. We weren't satisfied with this. So we went to a part 2 study. Oh, and let me just before I stop, let me stop for a second. For people who are like kind of chemistry buffs and things like that, with the hematoxylin and eosin, does anyone have an idea of maybe why we're getting lower RFU values? Well, I mean, it's believed that the hematoxylin actually doesn't completely dissolve, perhaps, and that it's somehow inhibiting the replication. That's a possibility. The eosin is very acidic. You can get maybe degradation there. And then when you're talking about Christmas tree stain, well, the nuclear fast red, my understanding is that it's probably not having any effect, both in the literature and in our own tests. But you look at the picro indigo carmine, it's very acidic, and also you've got indigo in there. And I think some of you know indigo has been as a known inhibitor. So here's part 2. Okay, we did an HE modified. We thought, well, maybe if we shorten the exposure times of these cells, it'll help. Because we liked that stain, but we just thought, well, maybe we'll shorter exposure times. And I do have all the protocols. The protocol that, let's see, actually I don't have the protocol in there, but in the final report is the protocol, which I'll give you the website too. I think the exposure times were down to about like 5 minutes and 30 seconds. 5 minutes for hematoxylin and 30 seconds for eosin. And then we tried just nuclear fast red alone. We thought, well, maybe that'll be good enough to ID the sperm. Then we tried a couple fluorescent stains, Cyber 14 and propidium iodide, which is used in a lot of fertility studies. And then I kind of threw in some other ones, this PSA, PI. PSA is actually a P-lectin, and I apologize, I don't know a whole lot about it, and we didn't go into detail. We didn't really have time to go into detail with this study. Don't know if it's appropriate for forensic samples. Again, that one's used mostly in fertility studies. And you'll see those. That's the protocol that you guys use today, and you'll actually see those samples later. So the evaluation, okay, were we able to evaluate, ID the sperm? We didn't do as much of a rating scale. We just kind of looked at them to see, can we ID them? But this time we collected the cells, and they were isolated with Lysin-Go extraction. By this point in our research, when we did this, we found the Lysin-Go was better for our work. And I will have a whole other presentation on Qiagen and Lysin-Go later, if you're wondering why we switched. And again, we performed the STR analysis for using Profiler plus loci. Okay. So looking at the just the bright field stains, we collected 300 sets of 300 sperm on the left, and we also collected sets of 300 epithelial cells. And here, again, it's the total RFU values obtained, unstained, H&E modified, and nuclear fast red. And there's, even though you see there's differences here, there's actually no significant difference between any of these values, either for sperm or epithelial cells. So that was great news. Just modifying, doing shorter exposure times using H&E, the same procedure, just shorter times, really helped a lot. Something to consider if you decide to go with the Christmas tree stain, even though you may get lower RFU values, you may think about lowering those exposure times might help, or changing your method of staining. So in part 2, we determined, I didn't have a chart for the Cyber-14 propitium because we got no STR results. Again, it's Cyber-14, I believe, is an interchelator, so that's most likely the problem. But H&E modified and the nuclear fast red protocols, they were good for sperm ID, and there was no significant difference in RFU values obtained compared to that of the unstained controls. So we were really happy that we had found a couple of different stains that we could reliably use. Again, PSA, FITSI, PI, the PI's propitium iodide. We just did real brief testing studies on that. It seemed like it kind of had very pretty dual stain. We couldn't get the consistency right in the beginning, and there was a lot of optimization that we still needed to do, and we didn't have time. But you did it for the class so that we could actually test the fluorescent capabilities for tomorrow. And this is actually, do you want, oh, actually I can't, can I hit these lights for a moment? Will that work or not? Yep. The big lights, can they go down for a moment or not? Just for a moment, yeah. So this is on a pen slide, and we'll talk, tomorrow they'll talk about autofluorescence of the pen membrane on a pen slide. So you'll see see if this will work on a dark background. Okay, so there's like, you can see the autofluorescence of those pores. But here are your sperm cells. And then this is the big epithelial cell there with the cell nuclei. That's kind of what you'll see tomorrow. Okay, you can turn the lights back on. Okay. So we'll move on to the isolation study. What did we want to get out of the DNA isolation study? Well, we considered a couple things. We wanted a real low manipulation method. Because in the back of our minds the whole time was low copy number. We then wanted to work in a small volume because these are little tubes and we wanted low manipulation. We wanted to kind of have a small volume format. But we also were concerned about purity. Do we need to purify these samples well? Because of the histological agents, because of other things, things to consider. So we looked at a couple things. Lysin-Go, which you use today, commercial lysis buffer. Very low manipulation. And it involves a series of heating and cooling to release the DNA and inactivate nucleases. Microlysis works, these are proprietary, so I actually don't know what's in there. Another commercial lysis buffer, also low manipulation, same kind of concept of a series of heating and cooling. We also use the QIAGEN kit that binds the DNA on a column, and then you clean it out, and then you release the DNA, and you have a very pure sample. We also experimented a little bit with Helix. We abandoned that fairly quickly because in the small volume format they were interested in, we're having a lot of trouble using Helix and kind of separating those beads out. If we got one bead in our mix, that was bad. And I know a lot of people are using that. We're hoping that it works, but at least in my protocol, it doesn't. And just to be aware that we added DTT to all of these protocols to lyse the sperm. So we collected sets of 300 sperm cells and 100 epithelial cells, and this compares the 3 different methods: microlysis, lyse and go, and QIAMP. And here on the left, the 10 loci you would expect to see in a profiler plus amplification. And microlysis right away performed poorly. And we got, we were lucky we get 50% of the loci showing up. But if you look at this, just the sperm cells in black, and you compare lyse and go and QIAMP, they all produced full profiles. We were happy with that. And then with epithelial cells, we also got fairly good results, not always 10 loci, and really no major difference between the two. But if we look at the next slide, then instead of just seeing, well, how many loci are we detecting, what's the amount of PCR product or RFU level that we're getting? And I crossed out microlysis because we've now eliminated microlysis because we can only get about 50% of the loci. Looking at the buccal swabs with E cells on the left, wow, you kind of look at that and you're like, wow, QIAGEN performed much better, had much higher RFU values than the lyse and go. It was actually a significant difference. But you look at the semen, and the lyse and go and the QIAGEN actually perform the same. There's no significant difference when isolating sperm cells using either method. So does anyone have an idea why that may be, why the different cell types behave differently? Maybe the cell size. When we were doing this experiment, first of all, we weren't just collecting the epithelial cell nuclei, we were collecting the entire cell body. That was just the protocol we decided to hit on. We're just kind of suspect that, okay, we're collecting the entire cell body, we're also collecting probably more cellular material, there's more mass there, maybe more histological agents. All this is going into one tube. Remember, lyse and go is an all-in-one tube process. It does nothing to purify the sample. So everything that goes in there stays in there. QIAGEN, on the other hand, is a purification process. So no matter what you do, you're usually removing all these impurities. And that's why we suspect that there was enough impurities and junk and cellular components in the E cells that it affected either probably the PCR process. But perhaps the smaller sperm cells, there wasn't enough of these inhibitors or things like that to affect it. So since our study was about collecting sperm cells, and we were looking for a method that was very quick and low manipulation, we went with the lyse and go for sperm cell extraction. Okay, so in summary, again, microlysis method not suitable for LMD with forensic STR analysis. QIAMP performed the best for collection of stained epithelial cells. We didn't really go a whole lot further with epithelial cells. That was not our focus. But this is what we determined in terms of the extraction. But both lyse and go and the QIAMP procedure performed well for isolating DNA from the LMD collected sperm cells. And what are the advantages and disadvantages? You have to kind of consider lyse and go, low manipulation, very inexpensive actually. QIAGEN, you get a nice pure sample, but there's higher cost, more manipulation. And so depending on what your sample is, maybe are you doing low copy number work, things like that, you'll have to kind of make a decision. And depending on your own extraction procedure, you would have to kind of run it through this to see, is it going to perform well? So then, let's go to the DNA yield study. Okay, Why did we do this? Well, we figured that the process itself of cutting out cells and counting them is actually a way of kind of quantitating how much is it going to eventually go into your reaction. But some things to consider, because I know that with DAB guidelines and things like that, validation, that you have to quantitate your DNA. That's always a quantitate, quantitate, quantitate. As a researcher, I'm not as concerned about that, but because I know the forensic community is, it's something that we have to kind of look further into. Is this in fact a method that you can estimate the DNA quantity? One thing you have to take in consideration, you can count the cells and estimate 3.3 picograms per sperm and things like that, but what do you end up with? You need to determine what's your extraction efficiency. And if you look in the literature, there's really no good source for and on the major extraction methods being used, what is the true extraction efficiency that you're getting? So we kind of looked into that so that we can kind of combine the two together to say, well, if we collect X number of cells and consider our extraction efficiency, this is probably what's going into the PCR reaction. We used two methods of DNA quantification when we collected LMD cells. And you'll see we use the QIAGEN protocol. The reason why, the real-time PCR method that we used, it's actually a method that's published. It came out of Vermont. Eric Buell is one of the authors. And he actually was, it was an NIJ-funded grant. We use that because it's extremely sensitive, more sensitive than any kit out there. And I could go down to 2 cells. We used that to quantitate our LMD samples. And we also kind of loosely made an estimate because we took those samples also and we did STR results. Kind of looked at the RFU values, kind of get an idea compared to some standards, known standards, to see where that fell. Okay, so the first panel here is of the sperm. And on the left is all the sperm, the numbers of sperm that we quantitated. And the middle column is by real-time qPCR. And again, the last column is just kind of by this just general estimate based on RFU values from our STR results. It looks pretty grim, doesn't it? Okay. Think, wow, I'm only getting 18%, 22%, in this kind of, but the thing that I noticed is that it's fairly consistent. It's not all over the place. Whether I collected 300 sperm or 5 sperm, I kind of got about the same amount of efficiency out of the extraction. Well, how do I know it's not the collection process as opposed to the extraction process? We also ran known quantities of DNA standards through the same extraction process. And surprisingly, we still it fell within this low range of extraction efficiency. So just running a known quantity of DNA through that extraction process, we got these lower results. The one on the left is also fairly consistent, but again, at least we were in the ballpark. We think, well, we were in the low 20s as opposed to maybe the teens. But I would say the real-time PCR results are more accurate. We also looked at E-cells, but we only did real-time qPCR on these. Well, gosh, somehow we had better yield on this, but again, fairly consistent. We have in like upper 30s and around 40% of yield of our DNA extraction process. And again, this was compared to, I think I don't have, I guess I don't have the DNA standards in there. But what do we kind of bring from all of this? Okay, it was surprisingly low. I actually was hoping these, especially a QIAGEN QIAMP, I thought that I would have a lot higher efficiency in extraction. And since there's no other publication regarding these efficiencies, I was like, well, I think that's kind of surprising to everyone. But the thing is, they're consistent between at least cell types. So looking at sperm or looking at epithelial cell, you can basically tag on an efficiency. And this is something that, depending on the extraction method you use, you probably will want to do something like this. So we can count cells using LMD. And based on the number of cells, we know how many picograms per cell, and factor in extraction efficiency, we can have an estimate of what's going in the PCR too. Now, most of in the work I did, it actually was really irrelevant because you really you amplify everything. You don't want to collect 1,000 cells. That'll take a long time. You don't want to collect 1,000 cells and dilute it, and quant it and dilute it. The idea is you just want to collect the right amount that you need to amplify. So what's nice is that you don't have to quantitate, at least in my mind, as a researcher. Validation and DAV is another thing, and I think we'll talk about that a little bit later. But the labor of going in and quantifying and consuming another part of that sample can be eliminated when you use laser microdissection.
That's a question, if you don't mind.
Oh, yeah, go ahead.
Did you do the same kind of comparison with in quantitation with the single tube extractions, where one wouldn't think you would lose DNA to manipulations like Kaizen washes?
Yeah, oh, I'm sorry. No, the reason why you were asking, like the Lysenko procedure that you're running today is all of a single tube process.
You haven't lost any.
Right. We originally, that was our goal. We wanted to do that test using that process. Unfortunately, we came to the determination after several tests that the Lysenko buffer is an inhibitor. Something in there, unfortunately it's proprietary, something in there is an inhibitor to qPCR that doesn't affect Amplistar amplification. And I know I've heard that some
does or doesn't affect?
It doesn't seem to affect the Amplistar reaction. But when you're talking about doing a real-time qPCR reaction, it seemed to be, that reaction seemed very sensitive to that buffer. And we were getting a lot of inhibition. And so we could not use that as kind of a gauge. So that's why we went to the Kaizen to kind of just get an idea of what we were collecting.
Did the epithelial cell,
no, go ahead.
The epithelial cells, you wouldn't necessarily have to use DTT with Lysenko, maybe.
Right.
I don't know, would that inhibit? Or have
I've heard it at certain concentrations it will.
Yeah.
Yeah, it will. I was using about 30 millimolar. And just be aware that even though we see these low extraction efficiencies, remember we're still getting profiles. It's kind of amazing almost. Okay. All right, now we're going to the mixer study, and this is what the meat of the whole purpose of this. We had to do all this workup before we got to, can we actually separate these samples from a mixture? So in the early studies, we were just kind of doing these general mixtures where we had one female oral cotton swab and 1 microliter of semen that we prepared. And we wanted to collect a lot of cells. We wanted to make sure it worked. So we collected 75, 150, and 300 sperm cell sets of those. And they were all recovered from these mixtures. PCR amplification was performed using very standard procedures, like right from the ABI user manual, 50 microliter reaction, 28 cycles. We didn't want to deviate from what maybe would be normally used in the forensic community. But we kind of added this little twist. We also, after we did that amplification, we took half of that and we amplified it for another 6 cycles using some more TAC. I did that not so much to increase the sensitivity of the sperm donor profile, but I did that because I wanted to detect if we had carryover from the female fraction. So this here is before any separation. We just want to see, in fact, did we make a mixture when we did this? And in fact, we had mixtures of the red asterisks are the female alleles, just to give you an idea. This is just one of the several mixtures that we prepared. So we took the sample and we collected 300 sperm cells. This is just one of the samples, separated by LMD, to see if we can just pull out the male profile. So in fact, this is entirely a male profile of the sperm donor that we used, and there's no female carryover. That was good news. And you'll notice the RFU heights on this. This actually improved after we got better at doing this. RFU values are a little higher. I have to say that I think these samples, I don't know Nick recognizes this at all. Nick, you ran these for me. So let's go a little lower, 150 sperm cells. On the left, using just the standard PCRs, we get the male profile. And I think there was just one in, we would occasionally on the 150 sperm cells, you see that next to the 10 is, there's one allele that wasn't called. It just fell below the RFU. So in that particular example, we just had one allele drop out. But still, a very nice usable profile available. There you see some imbalance a little bit, right there. Okay. So what happened on that extended? The reason why I didn't show the extended profile of the 300 sperm is because you do extended PCR on 300 sperm and you just blow out all your sample. It's just off scale. For 150 sperm, using extended PCR, we got the full male profile. So this is additional 6 cycles. And what I was happy to see is no female carryover.
How are these extracted again?
These particular ones, early on, were extracted using the QIAGEN QIAMP. Okay, so let's go lower. And I always like to show sometimes bad data because not all data is good. So 75 sperm cells. This is one of our worst 75 sperm cell collection data. Look kind of messy, the baseline. You have only amylogenin, and you only have a couple loci here that really came up. Not that great. I really put this up because what happens when you actually amplify this for more cycles, you get a full profile. And that told me that, wow, something about this is that we're looking into now the feasibility for this for low copy number work. To summarize kind of all the data, all the samples we did, under standard PCR conditions, using just 28 cycles, with the 75 sperm, we got about 71% of the alleles show up in general. 150 sperm, fairly good with about 95% alleles, and 300 sperm, all alleles would show up. Under extended cycles, no surprise, for the 75 sperm or the 150 sperm, we always would get all the alleles. Okay, so where is let me see the next slide. Okay, just kind of another way to look at data, because people are always interested in peak height ratios. The height of the lower peak divided by the height of the higher peak expressed as a percentage, because some people calculate peak height ratio a little differently, but this is my definition. So under standard PCR conditions, they all fall within acceptable ranges, kind of in this 80% range. Under extended cycles, not surprisingly, you're losing your peak height ratio at this lower level, 75 sperm. So the first part of the mixture study, we determined in our summary that LMD separation of sperm cells from an epithelial cell mixture results in a single semen donor. And the lower limit of detection using just those standard ABI protocols under standard condition, at that 75 to 150 cell range, you really kind of lose the sensitivity of getting a full profile. So what do we need to do? Well, to go lower, we need to extend the cycles to extend the limit of detection. That's where we go into our low copy number study. Let's see if I have more. Okay. So now we want to see, okay, at the low copy number level, how does the separation work? So these swabs actually is the same preparation. They're actually the same cell pellets that we used for the first study. But now we are collecting much lower amounts. We're collecting 80, 40, 20, 10, and 5 sperm cells by laser microdissection. And we did try 2 cells, kind of played around with that with no success. We didn't want to dive into solving the low copy number problem. We just wanted to see under these conditions what the limit was. So I didn't go any further with 2 cells. This time, Profiler Plus was done, but using 34 cycles and 38 cycles, straight off the bat. We didn't do this where we did it 28 cycles and we separate out the sample. I just did a straight 34 cycles and 38 cycles. And, I think all of you know the definition, low copy number. I mean, there are different definitions. Some people say it's below 100 picograms. Some say when, you're trying to analyze a sample that falls below a certain, perhaps peak RFU level, things like that. Okay, 80 sperm cells at 34 cycles. So I get a full male profile. And even better yet, no female carryover. Starting to see some imbalance, not unexpected. Here. 40 sperm cells. Also, the full male profile. Imbalance is getting worse. Here's some. Here's pretty drastic. But again, it looks very clean, doesn't it? I'm not getting any non-specific PCR product and no female carryover. 20 sperm cells. Again, same story, imbalance. And in this particular example, the full male profile and no female carryover. Getting lower and lower, 10 sperm cells, imbalance. And I don't know if in this one there's any dropout. There was sometimes dropout with 10 sperm cells, but in this particular example, there's no dropout of the male alleles. And then I don't have detail of the 5, kind of looking at each of the different amounts. It's like 80, 40, 20, 10, and 5. You can see that sometimes I get dropout here from the 10 cells. And here's one from the 5 cells. But what's great is that no female carryover. Now, okay, so you ask, I never got any carryover. I did once. And let's see if I put that in the next slide. We'll talk about that in a couple slides. So another way to look at the data at these 34 cycles. Okay, the percent of male alleles detected, this is all our data summarized. From at the bottom on the left is 5 cells all the way up to 80 cells. So if you're doing 80 cells and you're doing it at the low copy number level, you have no problem getting all the alleles. It's plenty of cells. 40 cells also pretty consistent. Almost, I think there was only one sample and a couple alleles that dropped out. 20 cells also look good. It's really when you get down to the 10 cell and 5 cell amount, so you really have a significant drop in the male profile and dropping out alleles. But they're still good partial profiles. So what about those 5 and 10 sperm cells? Can we actually do a better job in detecting more of the alleles? Well, that's why I went to 38 PCR cycles. You know what? Actually, let me back up for a second because I forgot one thing. You might be asking me, well, what do the RFU values look like? I don't have the values, but I will tell you the thresholds that I used. For 80 cells, all of the peaks were above 200 RFU. And for 40 cells, all the peak heights were above 100 RFUs. And when we get down to 20 cells, most of them were all 100 RFUs or higher. I think only just one sample. And then with lower than that at the 10 and 5 level, we get kind of in that lower level of detection. So let me start again here that if I boost it up to 38 cycles, what happens at this lower level? Well, we really determined that there wasn't much difference. You didn't really gain anything by going to 38 cycles, whether it's 5 cells, 10 cells. There's no significant difference. In fact, with using 38 cycles, we seem to find more non-specific PCR product. So 38 cycles was abandoned, and 34 cycles was found to be a good number to work with for a low copy number experiment. So coming back, okay, did I ever have a sample that I got female carryover? And I point this out because it did happen once. And the fact that it only happened once I was happy with, because I really do, as a researcher, consider it an outlier, but it's important to point out because it can happen. So one of the samples that I did, 80 cells and 40 cells, and funny that it didn't happen at the 20 cell and the 10 cell amount. probably because I was collecting less material and maybe that carryover wasn't as enhanced. But you'd see the asterisks are the female profile. Okay, now was this maybe it was just the technician wasn't very careful that day, things like that. So we went back to the same slide, we did the same thing, collected all the different samples, same results, got a mixture. In fact, I think it was like the technician did it and then I went back to the same slide, so 2 different people. Okay, we got another mixture. Then we went back to the pellet. Okay, this is something, the original pellet, re-smeared it, re-prepared it, did it. Then we didn't get a mixture, and we did it like twice. So we're thinking, we could never actually figure out why it did this. We just know that that particular slide seemed to have a mixture every time we collected from it. There's, you know, you can wonder like, well, what kind of environmental conditions were just right for that to happen? I really don't know. I mean, you can talk about maybe the exogenous DNA somehow having an affinity to the sperm head, and that's kind of something that's known that can happen even when you do preferential lysis and you have, a differential extraction, and you have tons of sperm and lots of epithelial cell too, and you keep washing and keep washing and keep washing. You don't see a single epithelial cell, but you still get a mixture. That is also kind of a theory that there's this free-floating epithelial cell DNA that's binding perhaps to the sperm head that you cannot see. It's just something to be aware of, and I don't know how this is going to translate into real forensic samples that are more aged and things like that. So the low copy number study in summary that minute numbers of sperm cells can be separated and recovered by LMD from epithelial cell mixtures. That's great news. And then STR genotyping can be obtained with as little as 5 sperm cell. And we were happy to see these nice, clean, whether they're partial profiles or full profiles of the male profile without the carryover from the epithelial cell fraction. And even also no non-specific amplification. And you start to think, well, I know a lot of people have, when they do low copy number analysis, a lot of times you get these, there could be dust and cells that are flaking off that you don't see that in a crime scene sample. And the thought is also, well, laser microdissection, you're smearing this on a slide. You're kind of avoiding all this floating debris because you're precisely dissecting. Perhaps that alone, you're avoiding some of these contaminants that have entered into the sample, or at least reducing the amount of contaminants that may have been in your original sample, that if you were doing a differential extraction would still all be carried along in the tube. Okay, let's see where we are time-wise. And I think I will go ahead because I want to try to get as much done as possible. We'll just go through the comparative study. So we want to compare how does laser microdissection perform compared to your differential extraction that you normally do in the laboratory. So we prepared these mixtures. We used hemocytometry to make sure we had all the right cell counts, and we prepared these precise cell mixtures from a 1 to 5, so 1 sperm cell to 5 epithelial cells, all the way out to 1 to 160. Took that pellet, and 9 microliters were smeared on the slide. Actually, there were the 3 holes with 3 microliter spots. And then 25 microliters went for a preferential lysis method or the differential extraction. We actually did not perform the differential extraction in our laboratory. I wanted to have that done by people who did it routinely. So a forensic lab did that. And so on my side here, on the left side, we did the LMD, collected, our target was 20 to 30 cells by LMD. We want to simulate the low copy number, whether there were a lot or not. We simulated this low copy number scenario, and we extracted with the lyse and go extraction method. And if you notice, you collected the sample in the tube, and it's going to be extracted in that same tube. And then the PCR master mix will be added directly to that tube. It will never leave that tube until it's ready for capillary electrophoresis. Preferential lysis, that laboratory did a differential extraction. They do an organic purification with Centricon. And then we qPCR'd. Those samples came back. We also did a real-time qPCR on those differential extractions to make sure what we had. And it ended up that all those samples, they were actually kind of on the low side, either was just at 2 nanograms if you did the maximum amount of input or fell below that. And these were all amplified at the same time, and capillary electrophoresis was done at the same time. at 34 cycles. So let's look at the easy one first, that each should perform well. On the left was laser microdissection, and the right is the differential extraction. So both performed equally well. They separated the male profile, and there was no female carryover. And that's no surprise. In the literature, a 1 to 5 cell mixture ratio, differential extraction should separate out the male profile. So we were happy, okay, that's a start. But let's look at the opposite end, the worst mixture at 1 to 160, and the story changes. On the left, LMD, the actual profile looks almost identical to the 1 to 5. It's still separated out the male profile with no female carryover, but the right is a different story. Using a differential extraction, the circles are the female alleles that carried over, and the arrows are the male alleles that dropped out. And you can see everything's at a very low level. So this is like, you can really see the performance difference between the two, especially when the epithelial cell density increases. So taking a summary of all the data, and take this first chart, basically says, how many % of male alleles am I detecting? So kind of the sensitivity. On the bottom is the ratios. And so ideally you want everything to be at 100. And if you look at the all the red boxes on top, that's the LMD. So it was at or near 100%, whether you had a lot of epithelial cells or very few. Not surprising, because I was collecting the same amount of sperm heads, either somewhere in the 20 to 30 cell range. But if you look at the preferential differential extraction method, the green circles, they performed very well when a mixture was just 1 to 5 or 1 to 10, but then it started deteriorating. As the epithelial cell density increased, you really actually could not pick up the male alleles very well. And this, again, it's very consistent with the kind of the ratio that literature reports on the limit of the differential extraction method. Okay, so now we just talked about, okay, the detection of the male alleles. What about carryover, the contamination? So ideally, we want to see in an ideal sample everything to be at the 0 range. And in fact, LMD, whether it's a 1 to 5 or 1 to 60, I wasn't picking up any female alleles. But using the differential extraction, somewhere around the 1 to 20, it starts losing it, and you're picking up more and more and more of the female alleles. So this is just a kind of a chart I threw in, a kind of another way to look at things. What about the amount of PCR product I obtained from this? The 1 to 5 and 1 to 10, they're very similar amounts. And again, there seems to be that 1 to 20 kind of barrier where everything falls apart for the differential extraction. But LMD still performs well. So in comparing the methods, it's kind of a workflow. So this is a little outdated. I have to apologize in terms of the time, because I know a lot of the methods have changed in the few years, and you've condensed some of and you've got automated systems, things like that. So using a differential extraction, okay, you do your microscopic ID, you're doing your preferential lysis separation. It takes 3 or 4 hours to do that, depending on your isolation technique. Could be just over an hour, it could be a couple of hours, depending on what method you use. And then you've got this quantification. Now, I know, though, this is old quantification numbers, 4 hours. I think now, what's more of the time frame for real-time PCR that people are using? What? An hour. And about an hour, okay. So that's more like an hour for more modern methods. And then you go to PCR and CE and data analysis. But on the right, okay, you still have to do the microscopic ID no matter what you do. But you can also collect the cells at the same time as you look for them. This is kind of an old 5 cells per minute with my kind of general average on the old instrument. Yours is faster, the one you worked on today. If you use a quick method like the Lysengo that I do, it's only a 20-minute, believe it or not. Those samples that you collected, all that is done is that DTT is added, and it goes through the incubation cycles. 20 minutes later, you've got your DNA ready for PCR. And no quantification. so not consuming sample. And so you do the same thing, but another thing to consider where you might save time is in the data analysis. If you are getting samples that are consistently more single source donors as opposed to mixtures, you're going to save time there and probably headache also, I assume. So the comparative study, and this is, I think, the last, we're going to kind of break after, we're going to end the class after this last part. So the LMD provides improved separation and detection from sperm, from epithelial DNA over a preferential lysis method like differential extraction, and particularly at these higher E cell to sperm cell ratios. And you don't need a quantification step if you use LMD, so you save time there. So if you think about it, if you actually do a single sample, you can really get that sample out quick, especially if you had a case that had to be rushed from just having the swab, analyzing it, and getting it on the CE. You can actually do it more rapidly. Pat's going to talk about that in much more detail about workflow. So this is just kind of a general thing. And I think I'm going to save the case studies till tomorrow, because that kind of, you're probably already tired by now, I'm sure. Was there any questions or comments? Something that you thought I might have missed?
You were talking about the DAB standards, and it's saying you have to quantify a forensic sample. How do you think that they would handle something like this by cell counting?
I mean, I think it's a discussion we'll also go into tomorrow, because I admit I haven't seen the new 2004 DAB standards because I've been out of the lab for a while.
It's still a temp quantification.
It's still a?
It's still a temp quantitation.
Yeah, and it's tough to say, and I think it also depends on your inspectors, things like that, because I know we did, even at LAPD, we did kind of a pseudo quantitation like this where we determined by our initial sexual assault screening based on kind of the cell counts, if we could just go without quantification do it, and they wouldn't buy that, doing any kind of cell count method. But it's something that we need to talk about, because as you all know, as technology improves, we need to make other changes. And is that one of the changes that need to be made in the standards, or is this going to be things like this where we start actually have data to back it up? Maybe do some real-time qPCR studies to show, and efficiency studies to show in our extraction, whether that is a good enough way to say we're quantitating.
It seemed to me that counting is quantitating.
Yeah,
As
and
long
that's
as you could identify them as human sperm cells.
right.
I mean,
yeah, are you gonna mistake it for a dog sperm
Right,
cell,
I mean,
things like that?
as long as you're convinced that they are human sperm cells, it seems like counting is quantifying.
May
to me.
I add to this? Because I've thought a lot about this, being that I've been a caseworking analyst now just about 10 years. Now, I came in right at the OCME. We were still doing PMDQ alpha, but then it was phased out about 3 months later. And I understand that standard was written because from the transition to RFLP to PCR, at first when people were doing PCR, they were using like all their extract. And then what was happening, of course, they were either getting blown out PCR, or the PCR was so saturated during AMP that what happened, you'd then get nothing, when in fact there may have been something there. So I think the standard was written that you had to do a quantification of your sample, so then you weren't wasting your sample. So of course my argument is I think the standard now needs to be revisited for newer technology, just like back then it needed to be revisited for new technology. And to me, one of the strongest arguments is I think this is a better assessment of quantity than the QuantiBlot was. I mean, QuantiBlot was not that great of an estimate when you get down to it. Now we're talking about real time and stuff. So if you make that sort of comparison, honestly, I don't know how they could.
But if you're going and you're consuming your whole amount that you're not catapulting or dropping into the PCR, and there was an inhibitor or something in your sperm, I guess whatever you were dropping, if you did the quantitation and you saw the inhibition, then you could clean up your sperm or whatever. I mean, I think you might miss that.
Yeah, it's it's kind of one of those things, like one thing, I guess you could, if you wanted to, you could try to do multiple samples. And it's something to consider. Are your forensic samples, like using a lyse and go method, are you going to have a high rate of inhibition because you're not cleaning up your sample? And you may have to go to a more purification method like HIAMP and see what, what rate are you getting inhibition? Is that going to be a problem? It's true, you could collect a sample, and how many microliters do you end up using for real-time PCR? Very small amounts. Yeah, 2 microliters. So it's still, you could, I'm saying you could incorporate that into your protocol, still do the quantification of laser microdissected cells to see if that's a problem. Pat, you want to?
I wanted to add that the standard, I don't think says you have to quantify the DNA. It simply says you have to assess the quantity of human DNA. It doesn't say you have to quantify to determine the amount of human DNA, just simply assess it. And I'm a firm believer that counting these cells is a method of assessment. Going back to the inhibitor, we've often thought about this, and the fact is you're only cutting out very small portions of it, which really should dilute the inhibitor if the inhibitor is a common material in there. But as we're going to see tomorrow, if this doesn't work, you can still use the whole spot anyway. So you haven't entirely consumed your sample in this method. Plus, you may have other samples in a rape kit that you can use. This is not like, just because you've cut 20 sperm up there does not mean there's only going to be 20 sperm on the whole slide or 20 sperm on another swab in the sample.
Right, I think, oh, I'm sorry, Soren.
It's okay, go.
I think that opens, a whole nother ball of wax, which maybe we can make part of our roundtable. But yeah, I think part of this case workflow thing is going to be somehow the slide will have to be assessed, period. Not just for quantity, even before you cut. I think somehow part of the protocol in your lab is going to have to be, okay, whether you implement Christine's procedure with we may still do that, where you make 3 spots, 3 microliter spots, and you look at, one or all of them to decide, well, is there enough even sperm here? Are there sperm here, and are there enough to do? If I needed to go back to that, could I go back and make more cuttings? If I lost the tube by flipping, if I, if I get a flat line, sample, it's a whole nother ball of wax. But yeah, these are things and I've tried to write a lot of these down that maybe we can talk about like in a roundtable discussion
tomorrow,
kind of thing.
Yeah. Any more questions or comments? Okay. Do you know when the shuttle is supposed to be?
5 o'clock.
Okay. So we're kind of.
Carl. Oh, sorry. Did you have a question?
Oh, yeah.
Yeah, it was just a comment, actually. I would advocate the assessment of a DNA that we get through a number of cells, which is okay. But then what with extracellular DNA from debris that we are not aware of there? So I would my comment or recommendation would be just in this transitional period when we are talking how we are going to assess, I would just stick with the real-time PCR still.
So you're suggesting that early, especially in these early studies, that kind of in order to kind of prove
exactly
that perhaps we in the beginning, take that 2 microliters, at least set it aside or a small portion, so that we have that option, or go ahead and run the qPCR kind of for a validation.
Exactly.
I think with your instance, you have to optimize the qPCR. It's probably not the inhibitor, the buffer probably changed, and that's why the assay didn't work. You know what I mean? There's probably some salt in there
that's.
So you probably could do that. That's like a whole other project.
Sure. Yeah, I know. Lots of other projects.
Lots of other projects.
Yeah. Well, I did do early on, and this was before I actually really optimized my procedure. I did do something kind of like that, where rather than go ahead and amp, I took that tube and took 2 microliters and did quant, did the real-time PCR, and I actually got, I want to say it was 30-40% of what I was detecting at least. So that may be something I could go back and do now that I've sort of optimized our procedure anyway, or the procedure I hope we'll use. Maybe I could go back again and do a real-time thing, quant to see what's happening.
I think Carl wanted to.
Coming from a non-DNA scientist, I think laboratories that get negative quantification results may commonly forward those extracts for profiling. And many of those come out with profiles that are usable. Do you guys do that procedure? And what would be the percentage of negative quants that give you profiles that are usable and probative?
Ours?
No.
Depends how accurate your quantitation procedure is.
Right.
Exactly, that's my point. The quantification procedure, if it's not just an assessment, if it's actual quantification and we don't trust it, I do think the standard needs to be thought of and changed, and the interpretation of that standard needs to be improved.
So what you're saying is that even though the requirements are that you have to make some kind of assessment of the quantity, even if you get 0, you still go ahead with the analysis?
I understand some laboratories do that. I know my laboratory
And that's
didn't do it
okay,
routinely, but
according
they
to
did
the
it when the
DAE
case results.
guidelines.
Can they fix their quant assay?
The what?
Exactly.
The assessment of quantity is just this magic thing that some auditors say, well, the only way you can assess quantity is actually quantify. Some people have said, well, I'm going to evaluate a sample as a very low quantity sample. I know that a cartridge case is not going to have an extraordinarily large amount of DNA, so my assessment is there's going to be a low amount of DNA in there, and just run the whole extract.
Yeah.
And there are people that do that, and that's their assessment, and that's what they're calling assessment. In this case, if you're counting the cells, you are assessing it. You're not saying it's 100% accurate. You're simply saying, I assessed it. And there's people that have taken that same standard and saying it absolutely means you have to attempt a quantification. Mm-hmm. And I happen not to be one of those people, but there are some people that do that. I mean, I showed a robot at one meeting, and they said, well, you can't do PCR in the same place you do extraction. Well, how in the world are you going to do it on the same robot deck? You know, there's analysts. I mean, one person came talking and said, you cannot do, the standard C, you cannot do PCR in the same room that you do your extraction. And that's how they interpreted the standard. So there are other interpretations out here. And I think there needs to be a comment on this, as the fact that, yes, cell counting, microscopy gives you some exquisite sensitivity. You can find a single spermatozoa in hundreds and thousands of epithelial cells. As you saw, just a 1 to 160 you were seeing. But you didn't say how many, that's just what you mixed up, but if you had smaller than that, you could have probably found spermatozoa in 1 to 500.
Yeah, I would hope so.
And so you're going into a new area of sensitivity, a new area able to enrich. I think we used that the other day, to enrich your sample for spermatozoa. At some point, do you really need to quantify that sample if you can count the number of cells?
Not to beat a dead horse, but but then of course it goes to the issue of estimation. See, that's what the quantify did. It really only estimated. It was not very accurate, and it just gave us a good estimate so that we could continue on with approximately 1 nanogram of DNA for PCR. So again, the counting of the cells, even though yes, maybe we're not getting a full lysis of every cell or whatever in there before PCR, we're just assessing. It's an estimate to help us.
I mean, if you assess it, okay, at 50 cells, I get 98% of the allele calls. Isn't that a good assessment of the amount of DNA that's getting in there and how to handle it? If I say at 25 cells, I may need to take this into account. At 10 cells, I may need to increase my cycle number. Those are all methods of assessing it, and you have a cause and effect. I count this causes me to do this other procedure or something else. Do you not? Are the people that quantify, and if I get over 2 nanograms per microliter, don't you adjust your concentration for amplification? So you've got a number, now you affect your procedure by that. If I do 75 cells, can I do it this way? If I do 50 cells, can I do it this way? All you're doing is assessing to basically make sure that you don't waste your sample or not unnecessarily use it or throw it away based upon not assessing it at all.
Robert?
I think it's a matter of perception, because one of the reasons why you're supposed to quantify is that you establish a target template level during your optimization steps. And I think it's just a matter of perception. If you establish that 25 cells will reach that threshold level, then it's a moot point as to how you quantify, whether you count whether you use quantifier or guess the blot, whichever one it would be.
I think that's a good point. You know, you've established a threshold for where you get a certain result or a good result. And I think that's an excellent way of putting it. At 0.1 nanogram per microliter, I can use 10 microliters and I get the ideal result. At 50 cells, we get the ideal result.